Hey guys, welcome back to Bioinformatics with R. We are going to work on some next generation sequencing data. So we're going to do RNA-seq, which is a more present method of quantifying gene expression changes compared to microarray. As I said, microarray is still useful because there's a lot of public data out there, but if you're planning an experiment going forward, because the price has come down so much, RNA-seq is very valuable. The difference between the two, microarray requires you to know the genes beforehand. So what it does is it takes probes that you've specifically decided to put onto the microarray and detects changes in expression of those probes. What RNA-seq does is it takes every single RNA molecule in your sample and sequences them so you can identify novel transcripts, unannotated genes, and things like that that you would not detect with microarray. So what the benefits of that are huge for novel gene detection and things of that nature. So without talking too much about it, let's get into it. Okay, so first thing I want to point out here is that I'm doing this one in a markdown file. Usually I've just been running R scripts, but I'm doing it in a markdown file because I want to show you how to integrate images into your knitted markdown HTML file. So we'll be doing this in markdown. We have 4 packages that we need to install. EdgeR is our differential gene expression package of choice. We will also be doing DE-seq in another series of videos. You're all familiar with dplyr by now. Annotation DBI is to annotate our genes, and then this library previously for the RNA or the microarray with BRIC, we use the Arabidopsis database. Here we're going to be switching it up because I know y'all are probably sick of plants. Plants are, well, you know, as a plant researcher, I will admit plants can be boring to someone on the outside. So we're going to be doing mouse instead. So there's not a lot of human spaceflight data because since there's so few number of astronauts, there's privacy concerns about putting their genetic information out there for everyone to use. So we'll use the next best thing, which is mice, which there's been a lot of mice flown on the space station. This data you should have transferred over when you transferred all your files in the previous video. So let's start by making an R chunk, or R code chunk, and we're going to load our raw data. Let's close off our chunk just for the heck of it. So we're going to do read CSV, and you should have a file that is called GLDS -102 underscore RNA underscore seq underscore normalized underscore counts .csv. And we're going to say that the row names equals gene ID. So let's run this. Oh, so I need to set my working directory. So for you, it's desktop, classroom, my files. I need to pick my directory. Hold on 1 second. Oh, it's under Bioinformatics, scripts, mouse, EdgeR, and open. Okay. And I'm going to save this file in that working directory. I'm going to just call it mouse EdgeR. Okay. So let's try this again. So as you can see, when we get this error, an RT error means that it can't find it because it's not present. So it's likely that you didn't set your working directory to the correct place. So we have our raw data. So we have, these are kind of like our probe IDs from the microarray experiment. This is the gene ID for mouse. We have our experiment. As you can see, we have 1, 2, 3, 4, 5, 6 flight experiments denounced by this FLT. And then we should also have 6 GC or ground controls. So call names for raw data are, we're just going to call them ID. So if we run this, we've changed our, oh, what's going on here? Oh no, we didn't need to do that. All right, so now we're gonna create a group. So this is like our targets. So this is another way of making your targets file. You can just type it in. You don't have to have a CSV file like we did for the microarray where we had brick 16 targets. So I'm just gonna do 1, 1, 1, 1, 1, 4, 5. All right, so 6 1s and then 6 2s. So you're in group 1 or you're in group 2. Flight or ground. 1, 2, 3, 4, 5, 6. Okay. All right, so we have our group. So now, let's do call a new variable, differential gene expression, GLM, which is a regression type. DGE list is our function, which comes from the EdgeR package. And we're going to say counts equals raw data, and our group is equal to our variable group. Okay, so we've done a GLM, and so if you look at the structure, that's what str stands for, GLM. This gives us some information. So it's a differential gene expression list from the package EdgeR. We have our expression numbers. We have our this character class, which is our gene annotation. Our columns are our experiments. And so that kind of gives us a little idea what we have in our. You can use this for a bunch of different things. So if I do str group. You can see it says it's a factor of 2 levels, etc. So it just gives you some information. So keep is we're going to keep CPM, GLM. So we want to keep only the so we want to make sure that we get rid of all the rows that have nothing in them. So this command says to look at the row sums and keep stuff where we have an entry, we have at least 4 entries. DGEG, oh, those are lowercase. Okay. So you see we have 24,000 elements, and to start we had, oh, so we didn't get rid of anything, so everything had an entry in it. So you do that depending on how deep the sequencing is. You'll have really rare transcripts that you sometimes don't capture, and so this experiment must have had very good coverage, it's called, so we didn't end up having to throw anything out. Okay, so let's keep going. Keep. Okay, so okay, here we go. So we did get rid of counts. And so here we're just setting the variable of keep. So it's a large logical. So if we look at head keep, it's saying whether to keep it as true or false. And then what we're doing is we're saying, depending on true or false, which ones to keep. So we started with this 24,000 different observations, and now we're down to 13,600. So that's how we read it up. Got a little ahead of myself. Okay, so let's look at the names of DGE, GLM. So you see we have counts and samples, which is perfect. So let's run this again. Okay, so moving on. Hold on here, I'm kind of make sure I'm in the right spot. Okay, so we have, so if we look at head DGE GLM, you can see we have the samples. This is what we're trying to pull up. The group, the size, and the factors, right? So there's 7 more rows, so we'll have group 1 and group 2. So now let's fit a model. Enough playing around and looking at data. So the design of our model is going to be model matrix. As you can see, this is creates a design modeler matrix by expanding factors and a set of dummy variables. So what this is going to do is it's going to take our whatever we put into it and design something readable for EdgeR to interpret how to separate our control and our experimental data. So we'll do by group. So now we have design. So as you can see here, kind of as we did before, we have if you are group 1, you're a 1 and a 0. If you're group 2, you're a 1 and a 1. It's just the way that EdgeR and stuff reads it. So we'll put this in your code to actually look at it instead of clicking on it. So, okay. So let's do DGE GLM com disp, and what we're going to do is estimate GLM common dispersion. So this says it estimates a common negative binomial dispersion parameter for DGE datasets with a general experimental design. So you'll see where this goes in a second. DGE GLM design, and then we're going to say verbose. So it's going to print everything so we can see what's going on. So we see our dispersion and our BCV. We're also going to look at a kind of a volcano plot to see the dispersion of our differentially expressed genes. DGE GLM trend of dispersion is we're going to estimate GLM trend disperse. So estimates the abundance dispersion trend to give you a approximate likelihood. So without, I don't want to get too much into the stats because that is kind of its own course. I believe I'm going to do a statistical theory or a bioinformatics theory course in the future, and we'll, because it could be, that's a whole quarter worth of stuff. Tag, disperse. But I do encourage you, if you are interested in the theory behind some of these commands that we're doing, to do the to look into the EdgeR documentation, so you can get this. That's just the introduction. So you can get the PDF. Why does it just give me an introduction? You might have to Google it. Yeah, I don't want eye ranges. Oh, it's behind my picture here. I'm looking at the help viewer. But yeah, so if you're interested in more of the background of these things, I encourage you to look at it. Tag-wise, disperse, DGE, GLM, trend, disperse, design. What did I misspell here? Estimated, or no, estimate. Okay. So we're going to plot the BCV, which is DGEGLM tag disperse. Plot BCV. Oh, here it is. Okay, so this kind of gives you an idea of the distribution. So the variance in the so this is average log counts per million. So this kind of gives you a distribution of the variation between samples, and so you want to see something uniform here, which this is pretty decent. So that shows that between our biological replicates, we don't have a ton of mess, and that they're somewhat uniform. GLM fit DGE GLM tag disp design. All right, so now we're going to do a GLM fit. We're going to add to our GLM fit with this DGE GLM tag distribution. And then let's take the column names from, am I skipping around here? Oh yeah, column names from the coefficient of fit. So we see we have groups 1 and group 2, which is good. Let's create another variable called GLM LRT fit the coefficient of 2. So as you can see here now, we're fitting a different regression to our RNA-seq data. So we have our coefficients, which is our change in expression. We have our method, which is like a one-way type ANOVA comparing. Deviance, so kind of like a standard deviation, so a measure of dispersal. We have our design in here. We have our actual dispersion, samples, so it says what our factors are. So we have two, you're either a 1 or you're a 2. So we got a bunch of data in here now. So let's do top tags of LRT. Actually, we don't even need to do that. Ignore that part. TTGLM top tags LRT n equals inf. Lowercase. Okay, so this gives us some more information here. So we have, this pulls out our, let's do head TTGLM. Oh, we don't want head. Let's just do TTGLM. It's not going to pull anything out because it's top tags. All right, so if we open top tags here, you can see it's a table where we have log fold change, log counts per million. So that's how many times it mapped per 1 million reads. The p-value, whether it's significant or not. FDR, which is our multiple test correction, so false discovery rate. So this is our adjusted p-value. Our adjustment method is Benjamini and Hochberg. I think that's how their names are said. But it's a BH correction method. The test is a GLM. So I guess that's kind of our data that we're going to go with here. So if we look at class of GLM, it's going to probably just tell us again that it's a top tags. Yeah, top tags, and it's from the package R. So I'm going to create another. What's going on here? Oh, let's close this out because we're kind of getting into the too much in a chunk here. All right, so let's run a summary on DEGLM, and we're going to use the decide test. DGE. So as you can see, the decide test identifies which genes are significantly differentially expressed from an EdgeR fit object containing p-values and test statistics. So it's going to look through the table at those p-values and those the test statistics and the differential expression, log fold change and stuff, and then tell us which ones are significant or not, or pull out the ones that are significant or not. And we can adjust the p-value here. So 0.05 is kind of the normal standard, I guess, in biology. But if you have a messier system or a messier data set, you might want to change it to 0.01 or 0.1. Sorry, if you messier, you probably want to change it to 0.1, so you're out a lot more error. If you have a nice tight data set, maybe you want to be more stringent, you can do 0.01, etc. We're going to show that our adjustment is that false discovery rate. We're actually going to change this because I ran this before and didn't have as great as results. So we're going to because this is a spaceflight thing, we're going to allow more false discovery, just because I ran it once and we didn't get great results. So tags of GLM. We're going to say that the row names of this GLM tag disp, and we're going to say it's as logical. So as logical creates a test object, so true or false of DEGLM. I'm going to run both these. Oh. It's looking for summary, could not find decide test. Oh, wait. What am I doing here? Decide tests. There we go. Okay, so what decide tests do is it says, all right, we're going to go through your list and we're going to say how many are upregulated or downregulated, how many are upregulated, and how many are not significant in group 2 compared to group 1. And we also did tags GLM. So this just gives us all the tags for the significantly differentially expressed genes, right? So we only have 223, so if you add these two numbers together, you get the 223 differentially expressed genes. So let's do a plot smear of LRT. DE tags equals tags GLM. So what this does is this is usually called the MA plot. Sometimes it's kind of a volcano plot. So this is showing all of our log fold changes for every gene, and then things in red are the ones that are differentially expressed. And what you want to see is kind of an even distribution of the differentially expressed genes over the average log counts per million, because if you have a concentration in one area, something's wrong. So you just want to see a normally distributed, significantly expressed across your average log counts per million. So you're not getting more significantly expressed genes at low average counts, right? So you want uniformity in that. So let's do AB line. We'll skip that. Let's do hist2. So we're going to make another histogram, TTGLM of, or no, sorry, hits2. Hits2. We're going to do TTGLM, so top table GLM. And from that, we're going to pull out the table and the TTGLM table, FDR less than 0.1, comma. Okay. Okay, so what is this doing? So we're making so we did this top table down here where we got our tags for our different genes that were significantly differently expressed, right? So now we're going to create a data frame, and what we're doing is this TTGLM, so it's a large top tags, right? So we have a table within a table and some values within this table, and so it's kind of like navigating down directories on your computer. And so we're saying, okay, look at top table GLM, and then look at the table. So top table GLM. Now we open table, and then we take within there FDR. Select from the table aspect the false discovery rate, which is this one, and we take out only the values that are less than 0.1 false discovery rate for our p-value, and we put them into hits. So we look at hits, we have all the genes where our false discovery rate was less than 0.1. Okay, so now let's write CSV hits 2, and we're going to say mouse EdgeR results 0 versus 1.csv. Okay, so I ran that. So now in our folder, Bioinformatics, scripts, EdgeR. So now we should have mouse EdgeR, and we just we just created a CSV from that institute table. All right, so we're a half hour in already. We got a little bit to go, but at this point, I am going to end the lecture. It's some of these things, I kind of glossed over, and I want you, this is part of the method, but like I said, I don't want to get too much into the theory on it because I could spend, a couple hours in the theory, but I encourage you, if you're interested in understanding what some of these commands did that I just kind of glossed over, like the estimate, GLM, trended distributions or dispersals and things like that, to look up the R vignette. I'm actually, there is going to be on Praxis, a learning path called resources, where it's not required, but I'm going to put all these manuals for these packages on there, and so you can look more into, if you're interested, the actual background behind what some of these do. Some of the stuff behind the scenes of what EdgeR or DESeq or whatever package we're using are doing, I think are a little deep for learning for this introduction stuff. But like I said, I'll probably teach a theory course in the future if you're interested in that. But if you are interested, there's plenty of documentation online to dig deeper into what some of these functions are actually doing. All right, with that, we're at over a half hour. We'll continue on in the next one. We're going to end up getting to path view, so we'll do some actual annotation of things in the next video. So I hope you had fun. I hope you